I hope you have seen my previous video on carbohydrates and the citric acid cycle. In this video, I will pick up where I left off in my discussion of the electron transport chain. I will focus on carbohydrates and the ATP synthase protein. The details will be somewhat dry, so bear with me to receive tidbits on the origin of life. The ATP molecule powers all life, so I find its formation fascinating. I am trying to give you the details of how things really work, not hand-wave my way through these explanations. If some details are unknown to science or unknown to me, I will tell you the truth, and not wave away the explanation as many explainers do. If you can follow, you will have a better idea of how life works. If you can't follow or have no interest in the details of how life works at the molecular level, please skip ahead. The figure shows a representation of the mitochondrial membranes, parts and functions, such as the citric acid cycle and the electron transport chain protein complexes. If we do an accounting of where and how many ATP were produced between glycolysis and the citric acid cycle and electron transport chain partners, we might find up to 38 ATP molecules can be produced under theoretical conditions. In real-case scenarios, ATP production is rarely so efficient, and in fact, in many cases, the ATP produced must be used in the citric acid cycle just to transport useful molecules through cytoplasm and across membranes into other locations where they are needed to participate in the citric acid cycle. From our discussion of glycolysis, we know that 1 ATP is used up to jumpstart step 1 of glycolysis. A second ATP is used up to proceed through glycolysis step 3. 2 ATP are made at glycolysis step 7, and 2 more ATP are gained at glycolysis step 10. So, glycolysis alone yields a net 2 ATP and 2 NADH. Without the citric acid cycle, these 2 NADH could power a fermentation step and be oxidized back to 2 NAD+. Now, if instead the glycolysis occurs inside the matrix of a mitochondrion, these 2 spare NADH could be used in the electron transport chain to push 12 protons H+ into the intermembrane space, and result in the formation of 5 ATP molecules when these 12 protons H+ return to the matrix through the ATP synthase enzyme. So, in total, glycolysis of 1 glucose uses 2 ATP but yields 9 ATP for a net yield of 7 ATP molecules. How many ATP will be made from the citric acid cycle and electron transport chain is dependent on the capability of the ATP synthase enzyme. Theory says ATP synthase has an efficiency of 3H+ equals 1 ATP. Measurements say ATP synthase has an efficiency of 4H+ equals 1 ATP. So when NADH pumps 10 protons H+ across the inner membrane into the intermembrane space, those 10 protons H+ only release 2.5 ATP when they come back through the ATP synthase into the matrix fluid. 1 FADH2 only pumps 6 protons H+ out through the inner membrane and back in through the ATP synthase enzyme. These 6 protons H+ give 1 FADH2 the value of 1.5 ATP. So, for accounting purposes, 1 NADH can yield 2.5 ATP and 1 FADH2 can yield 1.5 ATP. As was discussed, the 2 pyruvates can enter the citric acid cycle by different paths. The acetyl-CoA path or the oxalacetate path. If the 2 pyruvates produce 2 acetyl-CoA, and the reaction yields 2 NADH, and so can produce 5 ATP. As 2 acetyl-CoA are produced, each step of the citric acid cycle is doubled. If there is an absence of oxaloacetate, 2 pyruvates must produce the 2 oxaloacetate necessary to bond with 2 acetyl-CoA to yield 2 citrate. The production of 2 oxaloacetate by pyruvate carboxylase uses up 2 ATP. The 2 acetyl-CoA must be available from 2 other pyruvates which we just stated would produce 2 NADH or 5 ATP. So, in the absence of oxaloacetate, the case has a net yield of 5 minus 2 equals 3 ATP. So, Production of 2-citrate from 2-acetyl-CoA plus 2-oxaloacetate yields a net of 3 or 5 ATP, depending on the path of the 2 pyruvate molecules. We said that 1 NADH equals 2.5 ATP and 1 FADH2 equals 1.5 ATP. We said the citric acid cycle runs twice for each glucose. Inside the citric acid cycle, 1 NADH is produced when isocitrate is converted into ketoglutarate. 1 NADH is produced when ketoglutarate is converted into succinyl-CoA. The conversion of succinyl-CoA into succinate yields a GTP, which then transforms into an ATP, or double it to get 2 GDP, equals 2 ATP. The succinate conversion into fumarate yields 1 FADH2 as a result of the production of ubiquinol QH2, or double it to get 2 FADH2 or 3 ATP. The production of oxaloacetate from malate produces 1 NADH. If we add the NADH, we get 3 NADH, or double it, and we get 6 NADH, or 6 times 2.5 equals 15 ATP. Add 3 ATP from the 2 FADH2, and add 2 ATP from the 2 GTP. With simple addition, 15 plus 3 plus 2 equal to 20 ATP. So, running the citric acid cycle twice yields 20 ATP. When we add the glycolysis yield of 7 ATP, plus the pyruvate path production of 3 or 5 ATP, plus the citric acid cycle yield of 20 ATP, we get a total of 7 plus 5 or 3 plus 20 equals 32 ATP or 30 ATP. Not the 38 ATP predicted in theory. The difference is because the theory uses NADH equals 3 ATP and FADH2 equals 2 ATP. If we use the theory ratio of 1:3 for NADH to ATP and 1:2 ratio for FADH2 to ATP, we redo our accounting, we get the 38 ATP. The 2 NADH from glycolysis, plus 2 NADH from acetyl-CoA, plus 6 NADH from the citric acid cycle would be 10 NADH, or 30 ATP. The 2 FADH2 gives 4 ATP. Add back in the 4 net ATP from glycolysis, and we get 30 plus 4 equals 38 ATP. So, that is where the theoretical value of 38 ATP comes from. All of this bounty in ATP is a result of the citric acid cycle and electron transport chain that enable mitochondria and other aerobic bacteria to oxidize glucose into carbon dioxide and water. The overall metabolic reaction resulting from the oxidation of glucose is as shown. C6H12O6 glucose plus 6O2 reacts into 6CO2 plus 6H2O plus 32 ATP plus waste heat. For popular interest, many of the metabolites, enzymes, and coenzymes are used in glycolysis, the citric acid cycle, and the electron transport chain require precursor chemicals based on the vitamins we get from food or vitamin tablets. The various chemicals, particularly in the vitamin B family, participate in the citric acid cycle. Vitamin B1 thiamine is a precursor component in the formation of the enzyme pyruvate dehydrogenase that helps convert pyruvate to acetyl-CoA. Vitamin B2 riboflavin is a precursor component of flavin mononucleotide FNM and flavin adenine dinucleotide FAD. Vitamin B3 niacin is a precursor component of nicotinamide adenine dinucleotide NAD+. Vitamin B5 pantothenic acid is a precursor to coenzyme A, from which acetyl-CoA is synthesized. Vitamin B6 pyridoxine does not take a direct role in the citric acid cycle, but it plays a part in glucose storage, lipid metabolism, gene expression, and in hemoglobin synthesis. Hemoglobin is the iron compound in red blood cells that carries oxygen from the lungs into cells where the electron transport chain can use it. Vitamin B7 biotin is a component of pyruvate carboxylase, the enzyme that acts to convert pyruvate into oxaloacetate in the citric acid cycle. Vitamin B9, folate or folic acid, is important to the formation of DNA and RNA, and important to metabolism in that it activates vitamin B12. Vitamin B12 cobalamin has a wide variety of functions in metabolism and in the immune system. Specific to the citric acid cycle, vitamin B12 acts as a coenzyme in the formation of succinyl-CoA. For completeness, vitamins B4, B8, B10, and B11 have been used in the past to name organic chemicals that are no longer considered to be vitamins. Vitamin B4 adenine is a nucleobase. Vitamin B8 adenosine monophosphate is a nucleoside. Vitamin B10, aminobenzoic acid, is a precursor of vitamin B9 folate. Vitamin B11 tauroyl heptaglutamic acid is a growth factor. The vitamins K1 and K2, phylloquinone and menaquinone, have been identified in bacterial anaerobic respiration to help as electron acceptors for NADH and fumarate. In humans, vitamin K is most associated with blood clotting and bone formation. CoQ10 ubiquinone is not a vitamin, but a cofactor that plays an important electron transport role in the electron transport chain in aerobic respiration. There is no questioning the importance of vitamins and other cofactors in metabolic functions, including the glycolysis, the citric acid cycle, and the electron transport chain. If a human is deficient in any one of these vitamins, they will become sick. If a single-celled organism depends on these vitamins and cofactors for its metabolism, an absence of any one of them may cause the organism to go dormant or die. We can question how life came to find or produce or rely on vitamins for their metabolic function. Vitamins are not simple elements like other vital minerals like iron, zinc, copper, manganese, and magnesium. Vitamins are complex chemicals that can only be manufactured by living organisms. How vitamins came to exist is a subject of research. In the figure is an illustration of vitamin B2 riboflavin, the precursor of the metabolites FMN and FAD. It looks like three hexagonal benzyl groups joined together with some methyl groups, CH3, and some amino groups, NH, substituted onto the structure, along with other carbon chains with more hydroxyl groups, OH. The formula for riboflavin is C17H20N406. Alongside of riboflavin is the simple sugar ribose in its hexagonal carbon ring form. Riboflavin does not occur naturally. Animals cannot synthesize riboflavin. It must be synthesized by plants or fungus. Animals obtain riboflavin by eating plants or fungi or each other. Evolutionary biologists expect us to believe that a molecule as complex as riboflavin evolved from natural selection over 2 billion years ago. Maybe it did. None of us were there to see it. Pre-riboflavin plants or microbe fossils may never be found. Maybe I am wrong to question the evolution of riboflavin. It is just a question I have. How did riboflavin come into being naturally, and how did riboflavin come to be required for eukaryote metabolism in the citric acid cycle among other functions? In the figure is an example of vitamin B12 cobalamin. Among its many other functions, B12 is a cofactor in the formation of succinyl-CoA in the citric acid cycle. The B12 molecule is a knot of pentagonal and hexagonal carbon rings surrounding a single cobalt atom CO. The carbon rings have all manner of methyl groups CH3 and amino groups NH substituted onto the structure along with more hydroxyl groups OH and a phosphate group PO4. The formula for vitamin B12 cobalamin is C63H88 cobalt N14014P. Vitamin B12 does not occur naturally. It is synthesized by bacteria and archaea, including the gut bacteria, so a human or animal can absorb B12 from its own gut bacteria if it eats a normal diet. Vitamin B12 appears to be much more complex than the riboflavin that we just looked at. Evolutionary biologists expect us to believe that a molecule as complex as B12 evolved from natural selection over 3 billion years ago. Maybe it did. None of us were there to see it. The earliest B12-producing microbe fossils may never be found. Again, maybe I am wrong to question the evolution of B12. It is just a question I have. How did a molecule as complex as B12 come into being naturally, and how did B12 become required for so many life functions? We may never get an answer to these questions. I also have questions about the evolution of the electron transport chain. The protein complexes 1, 3, and 4 act as proton pumps from the matrix fluid into the intermembrane space fluid. The ATP synthase allows the protons H+ to re-enter the matrix from the intermembrane space while harnessing the pH gradient energy to make ATP. Were all these protein complexes formed separately or simultaneously? Did ATP synthase evolve before or after the protein complexes? There is an answer to the second question in existing primitive bacteria. All living organisms have some form of ATP synthase. In his article, "Is ATP synthase found in all life?" Brian Thomas writes, "If any bacterium is discovered without it, I would like to know about it. As far as I know, no one has come forward with a life form that does not contain some form of ATP synthase. And although living things have a form of ATP synthase, not all living things use the ATP synthase structure for making ATP. Some fermenting bacteria use only substrate-level phosphorylation to synthesize ATP. They make ATP anaerobically in their cytoplasm. They have ATP synthase for a different purpose. Proton regulation. They use up ATP to power the ATP synthase in reverse. Or they install the ATP synthase mechanism aimed outwards so that it works as a proton pump to expel excess protons H+ from their insides. If they did not, their cytoplasm would become too acidic and begin to destroy parts that they need to live. If any organism is unable to regulate its internal acidity, it will die. ATP synthase is one of nature's most unusual structures. ATP synthase is a biological rotating motor. It rotates. ATP synthase, bacterial flagella, and the rotors of gliding flavobacteria are all examples of rotating motors in a biological system. These three rotating biological systems may have a common origin in the design of ATP synthase. Flagella and gliding rotors may have evolved from ATP synthase. As all living things have ATP synthase in some form, it must be one of life's oldest, most ancient structures, maybe over 4 billion years old. Luca, the theoretical last universal common ancestor of all living organisms, must have had ATP synthase in some form, and passed instructions for building ATP synthase onto all its ancestors, everything alive today, from extremophile anaerobic bacteria to human beings. We all inherited a form of ATP synthase from Luca, or so the theory says. As shown in the figure, ATP synthase is a horribly complex, multi-part protein assembled out of at least 2,000 amino acids and probably more. Details of the structures of ATP synthase may give us a fuller understanding of how this wondrous mechanism works. ATP synthase has two main structures, F0 and F1. These structures have 6 subunits: 1 alpha, 2 beta, 3 delta B arm, 4 subunit C, 5 subunit A, and 6 the gamma camshaft or tail of subunit C. F1 is a non-rotating part, while F0 contains both rotating and non-rotating parts. F0 is made up of subunit A, subunit C, and its gamma camshaft. Subunit A is a stationary hollow cylinder part that is fixed to the inner membrane of the mitochondrion. Subunit A acts as the bearing in which subunit C rotates. The camshaft extends from subunit C and rotates inside the alpha and beta structures of F1. The gamma camshaft is bent, and the tip of this bent camshaft causes it to act like the cam lobe that opens and closes a valve in a reciprocating engine as it rotates. In the case of ATP synthase, the valves that open and close are the beta units of F1, the beta units slide back and forth against the stationary alpha units of F1. The alpha units are fixed to the delta B arm of F1. The delta B arm extends to subunit A, where it is fixed to the membrane so that F1 does not rotate with subunit C or its gamma camshaft. ATP synthase is found in mitochondria and gram-negative bacteria and in some form, all life. The central stalk is the gamma camshaft, as can be seen in the right side views. So we need to ask, why does subunit C spin around? What drives it? The simple answer is the proton flow across the gradient between the low pH intermembrane space and the high pH matrix fluid. Upon close inspection, we will find the rotation is all about the attraction of positive to negative charges, in the same way an electric motor works. When we look closer at subunits C and A, we find that rotating subunit C has amino acid cords stretched between its top and bottom rings around its outside perimeter. At the middle of each subunit C cord is negatively charged amino acid, aspartate minus, or the negative ion form of aspartic acid. Around the inside perimeter of the subunit A bearing wall are posts which are tipped with the positive charged amino acid arginine plus. When a proton H+ is pulled into the subunit C from the intermembrane space, it gets stuck to the negatively charged aspartate. There is still a negative charge on the aspartate, so it gets pulled towards the positively charged arginine on the post mounted to the wall of subunit A. As it draws closer, the positive charge on the arginine knocks the proton H+ loose from the negative aspartate. The proton H+ continues to follow the pH gradient out of subunit C and into the low pH matrix fluid. When the proton H+ releases from the aspartate, the full electric force of attraction between the negative aspartate and positive arginine is realized, and the force on the aspartate minus's cords pulls the whole subunit C to rotate into position aside of the arginine post. The momentum carries bare aspartate minuses past the post plus, just in time for the negative aspartate minus to capture another proton H+, falling through the pH gradient from the inner membrane space. As the bare aspartate minuses is catching the next proton H+, a new corded aspartate proton H+'s combination is rotating towards the positive charged arginine post. As it draws too close, the positive charged arginine pops the next proton free, and the force of attraction between the positive corded aspartate minuses pulls it towards the positive post-mounted arginine plus and rotates the subunit C towards the post, and the process of catching, pulling, releasing, strongly pulling, rotating past, and catching repeats. I did my best to try to represent this process in the figure. There is an excellent animation and explanation of this catching, pulling, releasing process of an ATP synthase motor on the YouTube channel The Glorious Clockwork between 5-minute 50-second and the 10-minute 50-second mark at the website shown. The YouTube channel Harvard X has 2 great videos, one of the entire electron transport chain and a second video focused on the spinning ATP synthase. See the links. Although the rotation process appears slow in these animations, in reality, the subunit C and its camshaft are rotating very fast, between 2,000 and 15,000 revolutions per minute, or between 30 and 250 revolutions per second. To get that fast rotation speed, ATP synthase requires between 300 and 2,000 protons to pass through it each second. Every rotation requires 10 protons. The fast rotation speed allows one ATP synthase motor to produce between 100 and 600 ATP molecules per second. The body needs that much ATP. As a rule of thumb, the human body requires its own weight in ATP every day. A 70 kilogram or 155-pound human uses 70 kilograms of ATP every day. The weight does not disappear. It just becomes transformed into the same weight in ADP plus phosphate. The ATP synthase reuses the ADP plus phosphate to make new ATP as fast as old ATP is broken down. Every cell's energy need is different. Liver cells have 2,000 mitochondria. A white fat cell may have only 10 mitochondria. Red blood cells are the only cells in human body that have 0 mitochondria. That said, not for scientific accuracy, but just for fun, we can play with averages. These averages are not for scientific use, but just for fun. If one imaginary average cell needs 10 million ATP per second, and one ATP synthase molecule can produce 100 ATP per second, then an average cell needs 10,000 ATP synthase motors. If the average imaginary cell contains 1,000 mitochondria, then each of these imaginary mitochondria needs at least 10 ATP synthase motors. So, how does the ATP synthase use the rotation to produce ATP? Once the subunit C and its gamma camshaft are rotating, the ATP synthase has the power to make ATP. The camshaft works by using its cam action to push out the beta units as it passes them. In the F1 structure, there are 3 stationary alpha units and 3 movable beta units wedged between those alpha units. As the cam tip pushes out a beta unit, a gap opens up between the beta unit and adjoining 2 alpha units. When the gap opens, a previously assembled ATP slips out into the matrix fluid. An ADP and a phosphate moves into the gap. Specially crafted molecules along the inside edge of the beta unit grab onto the ADP and phosphate with hydrogen bonds. The specialized molecules precisely line up the ADP and phosphate for the squeeze play that will force them together. Once the tip of the camshaft rotates past the beta unit can wedge itself once again between its adjoining alpha units. The closing of the gap between alpha and beta mechanically forces the third phosphate onto the ADP, and so it forms into an ATP. The camshaft rotates another 60 degrees to begin pushing out the second beta unit. As the gap opens between the second beta unit and its 2 adjoining alpha units, the process of releasing an ATP and bringing in another ADP plus phosphate for its squeeze repeats. The same process happens at the third beta unit. Once the camshaft has rotated 360 degrees back to start, the first beta unit is pushed out again and releases the ATP that had been formed in the first rotation. In this way, the F1 unit of the ATP synthase is able to create 3 ATP molecules for every 360-degree revolution of subunit C and its camshaft. All this action in F1 is from the energy released by the cascade of 10 protons H+ through the F0 rotor. The rotary motion of subunit C can be caused to run in reverse if ATP hydrolysis is encouraged in the F1 section of the ATP synthase. If ATP is hydrolyzed in the gap between the alpha and beta unit, ADP and phosphate is released. The ATP breakdown causes reverse torque on the gamma camshaft, which in turn causes the subunit C to run in reverse. The reversal of subunit C draws protons from the matrix and pumps these protons out into the intermembrane space of the mitochondrion. If such a reversal takes place in a bacterium, the inside of the bacterium becomes less acidic, while the outside of the bacterium becomes more acidic. Low internal acidity is beneficial to the bacteria. In the cases of some bacteria, the only purpose of the ATP synthase structure is to pump protons H+ out of the bacteria to lower its internal acidity. In such a case, the proton pumping structure is called VATPase. In primitive fermenting bacteria with no electron transport protein complexes, the VATPase may be their only way to rid themselves of excess protons H+ that would cause acid to build up inside their cytoplasm. Some evolutionary biologists believe VATPase evolved first. It was only later, when the electron transport protein complex was developed, that pumped protons from the insides of cells. That VATPase, or the ATP synthase structure, was repurposed to make ATP from the rotary action, rather than to use ATP as fuel to drive rotary motion and pump protons out of the organism. ATP synthase is ancient and must have developed with the first life forms. Some evolutionary biologists believe the flagella that is used to propel some bacterium evolved from the rotating F0 subunits of the ATP synthase. It is easy to see the resemblance of the corkscrew-shaped flagellum to the bent gamma camshaft of the ATP synthase. The rotating subunit C mechanism for driving the rotation of both the VATP-ACE proton pump and the flagellum of bacteria are remarkably similar in shape and protein construction and trace back to similar DNA genes. A word of caution. Flagella of eukaryote, protozoa and sexual gamete sperm cells and other multicelled eukaryotes do not rotate and are not derived from ATP synthase. Eukaryote flagella are formed from bundles of modified endoskeleton microtubule proteins. Sperm flagella and other eukaryote flagella whip around as the result of the contraction and relaxation of dynein walking proteins. ATP synthase is ubiquitous. Unless there is some life form discovered that lacks a form of the ATP synthase structure, it should be firmly established that all life requires some form of ATP synthase. Even primitive anaerobic bacteria that produce all their ATP by sublevel phosphorylation use a form of ATP synthase, a VATP-ACE, as a proton pump to regulate their internal acidity. Even if somewhere an organism is discovered that does not have a form of ATP synthase, it cannot be doubted that ATP synthase is at least nearly universal. In the absence of such a discovery, we say that ATP synthase is universal. And if the ATP synthase enzyme is common to all life forms, then the protein is ancient. The most ancient firmly dated life existed 3.4 billion years ago. There is unconfirmed evidence for life dating to 3.7 billion years ago, and even one claim of evidence for life dating back to 4.28 billion years ago. By all accepted geological evidence, the Earth formed just over 4.5 billion years ago. When it first formed, the Earth was completely molten, just exposed lava, no solid crust, no liquid water oceans, no liquid water period. The late heavy bombardment of asteroids and comets did not end until 3.8 billion years ago. So any life that was trying to form would have been blasted out of existence by the latest extinction-level crash of a giant asteroid. The giant impacts would make the 6-mile-wide asteroid that killed all the dinosaurs seem like a pebble. It was the comets from this late heavy bombardment that are credited with depositing most of the water that became our oceans. My point is, there is only a few hundred million years at most for life to form and the ATP synthase to evolve. ATP synthase requires several thousand amino acids to assemble. It is a horribly complex structure. ATP synthase is the only known biological structure that rotates other than the flagella and gliding rotary bacteria that probably evolved from ATP synthase. The big question I have is, how did the complex rotating and apparently necessary for life ATP synthase enzyme evolve in only a few hundred million years? Both the 3.42 billion-year-old accepted and the 3.7 billion-year-old disputed claims are most likely cyanobacteria that form stromatolites, ring pillars of rock. The cyanobacteria never went extinct. Living cyanobacteria still produce stromatolites today in isolated bays in Australia. These 3.42 billion-year-old and 3.7 billion-year-old cyanobacteria would certainly have had ATP synthase. If they are cyanobacteria, it means that ATP synthase is more ancient than 3.7 billion years old. The Earth was still molten 4.5 billion years ago. If the first life came into being between 3.8 billion years ago and 4.3 billion years ago, that would leave only 100 million to 500 million years for a fully developed ATP synthase to evolve. Look at the structure of ATP synthase in the figure. The F0 structure must rotate. How did that evolve in a few hundred million years? If the first ATP synthase enzyme was of the VATP-ACE type that uses ATP to pump protons H+ out of the cell, the F1 structure would have to evolve concurrently with the F0 spinning rotor. The best data ATP synthase I could find suggests ATP synthase is constructed of over 2,000 amino acids. That means over 10,000 nucleotide letters AGCT are required to code for the ATP synthase gene. How do 10,000 DNA letters randomly arrange themselves to code for DNA? A creationist can just say, God. I am waiting for an evolutionary biologist to come up with a credible answer to my question. I doubt they will answer my question in my lifetime, if ever. The fossil record is just too sparse. Scientists are lucky to find traces of life in rocks over 3.7 billion years old, never mind a missing link of a partially formed ATP synthase enzyme, or a 4 billion-year-old strand of bacterial DNA that they can sequence for the ATP synthase gene. The true origin of ATP synthase may remain forever unknown to science. The origin of the electron transport chain is as murky as the origin of ATP synthase. The electron transport chain is as complex as ATP synthase, although all 4 protein complexes of the electron transport chain did not have to evolve concurrently. I can see a life form having just 1 or 2 of the protein complexes to use as proton pumps as having an advantage over a life form that is only using VATPase as a proton pump to control its internal acidity. The electron transport chain is not as ancient as ATP synthase. The protein complexes of the electron transport chain must have somehow developed as alternative proton pumps to the ATP synthase-based proton pump VATPase. The organisms that developed these protein complexes as alternative protein pumps did so because the VATPase proton pumps use ATP to pump the protons out of the bacterial cell, while the protein complexes use the electron current generated in the electron transport chain to pump protons out of the cell. These protein complexes use NADH and FADH2 as a source of electrons and energy, as opposed to the VATPase proton pumps that use the mechanical breakdown of ATP into ADP in their F1 structure to rotate their F0 subunit C rotor to pump out protons H+. Once the protein complexes became efficient enough and sufficient enough to pump out all the protons H+ produced by the cell's other life functions, the VATPase was relieved of its proton pumping responsibilities and was now free to act as ATP synthase and synthesize ATP full-time. All the cell needed to do now was to harness free oxygen O2 in its ATP generating process to become a fully aerobic creature that breathes air or oxygenated water. Free oxygen O2 was waste produced by the photosynthetic anaerobic bacteria, such as cyanobacteria. Free oxygen O2 became plentiful enough for aerobic life around 3 billion years ago. Around 2.4 billion years ago, the atmosphere became so polluted with poisonous oxygen that 99% of all life went extinct. Only the fully aerobic life survived in the open. The anaerobic life survived this great oxygenation extinction only if they lived in deep crevices in the earth, or in deep ocean mud, or had thick cell walls to keep the oxygen poison from oxidizing their insides. It was during this 500 million year window, 3 to 2.5 billion years ago, that fully aerobic cells evolved along with the protein complexes that allowed it to make use of the available free oxygen O2. A fully aerobic cell was no longer dependent on fermentation to generate ATP. It could use the citric acid cycle, its protein complexes, and its ATP synthase enzymes to generate far more ATP for energy than its fermentation-dependent neighbors, and was able to use its extra energy to outcompete its fermenting bacteria neighbors for survival. Among the first bacteria to become fully aerobic would have been members or cousins of the alpha proteobacteria group. This transition from anaerobic fermenting bacteria to a fully aerobic cell must have occurred between 2.5 and 3 billion years ago. I don't know if 500 million years is enough time for life to develop the protein complexes for using oxygen. They did do it, and I will go through the possible stages of electron chain development at a later point. One of the fully aerobic proteobacteria was an ancestor of the mitochondria organelles found in cells of every eukaryote organism. At some point around 2 billion years ago, one microbe, thought to be an archaea, engulfed a proteobacteria ancestor of mitochondria. Instead of digesting it, the engulfing microbe found the proto-mitochondrion produced more ATP than it could otherwise obtain by digesting the protomitochondrion for food. The microbe incorporated the ATP-producing protomitochondrion into its cytoplasm. Scientists don't know if the engulfing and incorporation was a one-time event or if it happened multiple times. The engulfing microbe became the first eukaryote. Two different species developing a relationship for their mutual benefit is called symbiosis. A relationship which involves one species taking another species inside its body for the mutual benefit of both species is called endosymbiosis. The eukaryote host has an endosymbiotic relationship with its tenant mitochondria. Its mitochondria provides the eukaryote with ATP energy, while the host eukaryote provides the mitochondria with food, glucose and oxygen and a habitable environment, and, all important, protection from being engulfed by a foreign predator that might digest it. Both the host eukaryote and the mitochondria have their own separate DNA. The mitochondria divide and reproduce daughters that remain living within the eukaryote host. Now, 2 billion years later, eukaryotes cannot survive without their mitochondria, and the mitochondria have devolved into an organelle no longer able to survive outside of the eukaryote cell. The mitochondria's cousins, the alpha proteobacteria, still survive in the wild in isolated bodies of water. Who is to say which group is more successful, the wild alpha proteobacteria or the modern mitochondria living in comfort as long as its eukaryote host survives? In the intervening 2 billion years, the engulfing microbe species has founded 5 new kingdoms of life: animals, plants, fungi, chromists or algae, and protozoa. The organisms in each of these kingdoms are within the superkingdom or domain of the eukaryotes. Each of these eukaryotes are distinguished as eukaryotes by having cells with a distinct nucleus and mitochondria. Organisms lacking a distinct nucleus and mitochondria are in the prokaryote domain. Within the prokaryote domain are 2 kingdoms, bacteria and archaea. In the kingdoms of bacteria and archaea, all the members are microscopic single-celled creatures. Some eukaryotes still exist as single cells, the protozoa, and some algae. Only the eukaryotes have developed into multi-celled creatures. The eukaryotes were able to become multi-celled organisms in part because they can utilize the enormous amount of ATP that their mitochondria can produce. It would be impossible for bacteria to develop into multi-celled creatures without a means to increase their ATP energy. Plants and many chromist algae were able to incorporate cyanobacteria into their own cells through endosymbiosis, and so were able to become sugar producers through photosynthesis. The cyanobacteria incorporated within plants and algae have since devolved into chloroplast organelles that are no longer able to survive free of their hosts. Chloroplasts are the keepers of the chlorophyll molecules in plants that are able to conduct photosynthesis, create sugar from CO2, water, and light energy. It is believed that the endosymbiotic event to incorporate cyanobacteria into plants and algae occurred after the first eukaryotes incorporated mitochondria, because all eukaryotes contain mitochondria, while only plants and algae contain chloroplasts. The endosymbiotic event of eukaryotes incorporating cyanobacteria into their cells took place between 2 billion and 600 million years ago. So, if alpha proteobacteria and the ancestors of mitochondria developed the electron transport chain of the 4 protein complexes between 2.5 and 3 billion years ago, what might have been the intervening steps between nothing and the full electron transport chain? To answer this question, I looked up information on the electron transport chain on the National Institute of Health (NIH) website, and specifically at an article on the NIH website entitled "The Evolution of Electron Transport Chains" at the webpage on the slide. This article makes many references to the book Molecular Biology of the Cell by Alberts, Johnson, and Lewis. The NIH explains what we could already guess. As the Earth became more and more populated with fermenting bacteria between 3.5 and 3 billion years ago, the waste products of fermentation, namely protons H+, would have acidified the oceans. Modern fermenters excrete acids such as formic acid, lactic acid, acetic acid, and succinic acid, and these were likely the same waste acids produced by the archaic fermenters. Photosynthesis would have already started quite early with the cyanobacteria. Cyanobacteria are photoautotrophs that make their own food using light energy from the sun to split water and CO2 and recombine them into glucose. The waste product of the photosynthesis process is free oxygen, O2, which would bubble up out of the water into the atmosphere. That free oxygen is great for us and the rest of the modern aerobic life on Earth. But free oxygen is poison to anaerobes that populated the archaic ocean floor, including the cyanobacteria and other anaerobic photoautotrophs that were making the oxygen O2. As the oceans became lower in pH and more acidic, the archaic life would have had to develop tougher cell walls to keep out the acid or protons H+ they were expelling. The tougher cell walls might have offered protection from the free oxygen poison as well. Those archaic life forms that did not adapt to the highly acidic ocean's condition went extinct. About this time, 3 billion years ago, some of the archaic life began experimenting with newer forms of metabolism. Glycolysis with fermentation provides both ATP for energy and NADH as an electron donor. Most of the time, NADH is used to make more ATP. Archaic life might have explored other metabolic pathways and different uses for its available NADH electron donor. The NIH article suggests 3 stages of metabolic changes that might have led to the development of the electron transport chain. In stage 1, the rise in ocean acidity would have required increases in the use of the reversed form of ATP synthase. The ATP-consuming VATPase and its variants that act as electromechanical proton pumps to pump out more protons H+ from inside the cells and prevent the internal acidification that would kill the cell. The use of ATP synthase to pump protons H+ out of the cell uses ATP and reduces the availability of ATP for other cell functions. In stage 2, the archaic life would have tried primitive electron transport style methods to rid themselves of excess protons H+. The NIH article suggests protein enzymes might have been developed that would convert the available formic acid into fumarate. The conversion of formic acid to fumarate requires electron transport and produces the energy needed to electrically pump protons H+ out of the organism into its surroundings. The NIH points out that modern extremophiles that live on formic acid CH2O2 have such protein enzymes and use them to conduct such proton pumping actions. An example of the formic acid metabolism is shown in the figure. From the figure, we see at the cell exterior the formic acid break down into 2 H+ and CO2. The resulting 2 electrons are transported into the cell by the enzymic proteins and ubiquinone Q. Inside the cytosol of the cell, the 2 electrons are used with 2 different protons H+ to bond to fumarate to convert it into succinate. If the backflow of the protons H+ is prevented, there would be a net loss of 2 protons H+ inside the cell and a net gain of 2 protons outside the cell. Effectively, the formic acid metabolism acts as a proton pump. The formic acid pump uses no ATP, and so does not reduce the cell's store of ATP. Modern bacteria that live off energy from formic acid include strains of E. coli. The figure shows the reactions in modern formic acid metabolizing bacteria that uses fumarate to succinate conversion to pump protons. The example given in the figure is a graphical representation of formic acid metabolism that occurs in a real strain of the bacteria, E. coli, in a formic acid environment. The formic acid electron transport chain involves only 2 protein complexes with a ubiquinone intermediary. So, it is truly much simpler than the 4-protein electron transport chain plus ATP synthase in fully aerobic mitochondria of eukaryotes like us. A reasonable person would accept this formic acid electron chain example as an intermediate step on the evolutionary path onto the full 4-protein complex electron chain. On the other hand, a reasonable skeptic might ask the questions: how did the 2 protein complexes evolve? Which came first, the protein enzyme that accepts the electrons and breaks down the formic acid into protons and carbon dioxide, or the protein enzyme that donates the electrons to convert the fumarate into succinate? And how were these proteins coded into the DNA? By random chance? or by intelligent design. When I was in school, all professors lectured with a shortcut teaching technique called hand-waving. They would write down some preliminary assumptions, wave their hands about while talking, and then write down the result on the board, having skipped all or most of the steps in between the assumptions and the answer. It kept lectures within the time limits of the time allotted. We would all still be in school if every question needed every step along the way to the answer to be written out. In some cases, a bacteria generation can grow and divide every 20 minutes. There are 525,960 minutes in a year, so bacteria might go through 2,500 generations in a single year. So, in 500 million years, there could be 125 billion generations of bacteria. Are 125 billion generations enough generations for bacteria to develop the complete 4-protein complex electron transport chain by random chance? Some reasonable scientists say yes. The yeas might include Richard Dawkins, Edward O. Wilson, Carl Sagan, and Lynn Margulis. Some reasonable scientists say no way. These skeptics might include Stephen Meyer, Michael Behe, and James Tour. To continue our story of the evolution of the electron transport chain, in stage 3, bacteria that increased the efficiency of their electron transport chains for pumping out protons H+, would have a distinct advantage over bacteria that relied solely on their VATPase as proton pumps to lower their internal acidity. Those bacteria with the most efficient electron transport chain proton pumps would no longer need to use VATPase as a proton pump. These bacteria could reverse the flow of protons H+ back into their cytosol through the VATPase, and so begin to use VATPase as ATP synthase to generate ATP. These proton-pumping bacteria could allow their ATP synthase to take protons into the cell to generate ATP without causing their insides to acidify, because their electron transport pumps were so efficient at expelling protons. At first, these electron transport complexes might have used nitrite NO2- or nitrate NO3- as the electron acceptor. As free oxygen O2 became more available, the proton-pumping bacteria would have switched over to using oxygen as their electron acceptor because oxygen O2 is more reactive than either nitrite and O2- or nitrate NO3-. Modern bacteria that can use either nitrite NO2- or nitrate NO3- as electron acceptors in their metabolism include planktomycetes and Thiobacillus denitrificans. The equations show that NO2- reacts with 4 protons and 3 electrons to produce nitrogen and 2 moles of H2O. NO3- reacts with 6 protons and 5 electrons to produce nitrogen and 3 moles of H2O. O2 reacts with 4 protons and 4 electrons to produce 2 moles of H2O. The figure shows a stage 3 electron transport chain that can use nitrate NO3- as the electron acceptor. At stage 4, after 500 million years of development, the 4-protein complex electron chain plus ATP synthase was developed in wild protomitochondria. These protomitochondria were engulfed endosymbiotically by another bacteria, and the first eukaryote came into being between 2.3 and 1.8 billion years ago. The figure shows the final stage 4 is a modern electron transport chain that uses oxygen O2 as the electron acceptor and ATP synthase using protons H+ to create ATP. In my next video, I will discuss how photosynthesis makes sugar.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.